Good afternoon, and thank you very much for coming to TEDx The Brain. I hope your brains aren't full yet. I have the great fortune of working together with a fantastic bunch of people whose contributions I wanted to acknowledge at the outset. This is my lab, here they are. And I'm a social neuroscientist, so what drives my passion for the research that we do is a genuine sense of awe and amazement looking around at our social behavior. Pretty much everything that you see right now and everything you've seen today at this TEDx event is social in some sense. Obviously, watching me stand here, gesticulate, and talk to you is social, but so is TEDx, so is all the technology you see here, so is the construction of this building. All of that depends on our social behavior in some way or another. If you go outside and you walk around the streets in Pasadena, you realize we live in largely a socially constructed environment. Almost everything you see is in some way or another, directly or indirectly, a consequence of our social behavior. And this is amazing and I think unique. And I wanted to just emphasize this point a little further with the next slide. This is one of these wonderful images that you've probably all seen of the Earth at night, taken from the International Space Station. And surely, if you were an alien spaceship and you're approaching Earth for the first time, in addition to being dazzled by the beauty of our planet, you would be amazed at what you see here, this pattern of lights due to cities, highways between them, as well as satellites and the space station twirling around. We know of no other species on this planet or anywhere else that's achieved anything like this. We've completely transformed our planet, for better and for worse, as a consequence of our social behavior. It depends on culture, the transmission and accumulation of knowledge across generations, depends on very large-scale collaborative behaviors, and we know of no other species that does anything close to this. There are other species that live in very structured societies, for instance, use social insects like termites, ants, and bees. They haven't achieved anything like this, and certainly our closest living relatives, apes and monkeys, don't exhibit anything like this kind of behavior. So here's a deep puzzle, and as neuroscientists, we think that this must be reflected in some way, indeed can only be understood by investigating the human brain. So that's the basic motivation. What is it about our brains, about this chunk of tissue, 80+ billion neurons, each with about 1,000 connections that you heard a lot about in the earlier talks today, that makes all of this possible? And one answer is that our brains are big. Anthropologists have pointed out that our brains are much bigger than you would expect given our body size, about 1,400 grams or so for an adult human brain. By contrast, a chimpanzee's brain is only about a third that size, about 400 grams, about the size that our hominid ancestors would have had about 4 million years ago. And indeed, there's a correlation in primates between the relative size of the brain and the size and complexity of the social group that the animal lives in. So you need big brains to transact all the complex social behaviors that we do. That's fine and good. As neuroscientists, we're more curious, and we would want to know what it is about this big brain. What are different parts of the brain doing that mediate our social cognition and our social behavior? Hence, the social brain, the title of this talk. So what you see here, this is a collection of brain structures, all these different colors that you see depicted on various views of a human brain. And you should think of these not as operating in isolation. They're all connected with one another. They're a big network, a system for processing social information. Much of this knowledge has come from functional magnetic resonance imaging of the human brain. Some of it has come from studies of rare patients who have focal lesions, damage to particular parts of the social brain, and I'll show you some such examples in just a second. And we are also very interested in looking at differences in functioning of these social brain structures across different people. Atypical functioning in certain special populations that exhibit unusual social behavior, for instance, people with autism, whom we also study in my lab. Now, what are the social processes that I've been vaguely alluding to? Very roughly, you could think of partitioning these into three. So at the input end, there's social perception. So right now, you're watching me gesticulate and move around. You're looking at my facial expression. You're listening to what I'm saying. And so visual and auditory parts of your brain need to process that. And at the output end, there's social behavior. Interposed between input and output in the middle here is a set of processes, social inference, that goes beyond what you can see, beyond what you can hear, to make inferences about what you cannot see, namely the internal states of other people, how someone is feeling, what they're thinking, what they're intending to do next. These are inferences we make all the time based on observations of other people. So these two processes, social perception and social inference, are the two that my lab has been studying in some detail. And I just want to give you a flavor of them and the distinction between them with an example here. This is my mom and her pet deer. My parents live in a very rural environment. And to first order, my mom and the deer are doing very similar things at the level of social perception. My mom sees the deer's face, just as the deer sees my mom's face. But then my mom does a bunch of stuff that the deer doesn't really get started with. So the deer looks at my mom's face, smells my mom, and has a simple association to a forthcoming food handout. My mom looks at the deer and starts doing all kinds of things and thinking, oh, the poor thing, it's orphaned, it must be feeling very lonely, it looks like it's feeling cold, it's feeling hungry, etc. So that's what I mean by social inferences. It's something that we do all the time. How do we investigate this in the lab? Well, social psychologists looked at this and devised a stimulus, a little video that I'll show you in just a second, that we also used in our experiment back in the 1940s. Fritz Heider and Marianne Simmel wanted to isolate this process of social inference, and they did so by making this very simple video. I'll start it now. That has no faces. There's nothing overtly social here. There's no people. There's no soundtrack. And if you were a participant in our experiment, we would just sit you down and simply tell you, look at this, just watch and see what happens, no other instructions. And when it's over, just tell us what you saw. And what you see on the screen here are these three geometric shapes moving around, and they move around and do various things. The actual video is a little longer, a little slower, and you don't have me talking in the background to distract you. If you saw that, you would produce a description that runs something like this, that's notable for the fact that there's a story there. So people automatically imbue this with social meaning. They see a box, there's something going on, certain of these characters are being chased, some are scared, some are happy, and of course we can quantify these. So all these red words that you see here are words that are brought up by the social inference process, words about goals, intentions, emotions, and so forth. And we do this automatically. In a sense, you could think of this as a social illusion. It's like the illusions that Marcus Meister showed us, more complex, but it's a social illusion that illustrates our propensity to anthropomorphize, which is something we do all the time. We see faces in clouds, we get angry at our laptops, and you see social meaning in little geometric shapes moving around. Now, the interesting experiment that we did is we showed the same video, did the same experiment in a very rare patient known as subject SM, who has lesions, damage to a particular part of the social brain. She saw the same thing, and we asked her, what did you see? And she said the following, I'll just read it with you. She said, okay, so a rectangle, two triangles, and a small circle. Let's see, the triangle and the circle went inside the rectangle, then the other triangle went in, and then the triangle and the circle went out and took off, left one triangle there. And then the two parts of the rectangle made like an upside-down V, and that was it. Now, this would be very difficult for you to produce as a description of what you saw here. This is geometrically accurate. This actually happened, and it illustrates important points, which is that there's nothing wrong with this patient's vision. It's not that she has blurry vision. She saw what was happening. She encoded it into memory, and she could tell us about it. So perception, memory, and language are intact, but she fails to be able to link what she sees here with inferences about what she cannot see. Now, where's the lesion in this patient? It's that orange area on the far right of the social brain. These are two structures, one on the left, one on the right, roughly in where your ears are, a structure called the amygdala, which is Greek for almond, that my lab has studied in great detail. It's probably the structure that's been studied most with respect to social neuroscience. And it's ideally positioned to mediate this process of social inference that I was speaking of. So it gets input from high-level visual parts of the brain, like the face patches that Dorit Tsao told you about. It gets that as input, and then it's able to associate that visual information with value and with all the inferences that we make about that stimulus through its projections to many other brain structures, for instance, in the frontal cortex. There's a wealth of evidence implicating the amygdala in social behavior. For instance, if I scanned each one of you now in an MRI scanner and measured the volume of your amygdala, it turns out that the size of your amygdala correlates with how many friends you have. Social network size and amygdala volume are correlated in humans. I want to turn in the last minutes here to a second and very important part of our social behavior with another family picture from Christmas with my lovely wife, who's in the audience here, carving up a roast. And what's notable in this image is that everyone in this image is looking at a person. My wife is looking at me taking the photo. There's my two nieces. Older niece is looking at my wife. Her younger sister is looking at her older sister. And my mom, again in the background, is looking at my wife. And indeed, all of you in the audience spend most of your time looking at the faces in this image, as you're spending most of your time looking at my face as I'm talking here. And again, this is something we do all the time. We seek out social information in the environment because it's so salient. We look at other people, we look at their faces. But this suggests an important revision to this scheme that we had up here, which is that on the input end, social perception is not like taking a picture with a camera. It's not that you just open your eyes and all this information comes in and then your brain processes and does all the social inference. Instead, social behavior determines what you perceive in the first place. And of course, this happens at many levels. So how you behave influences other people's reactions, and you perceive that. But also where you look, where you direct your gaze and your visual attention, influences what your brain has to work with in the first place as the basis for the social inferences that it makes. How do we test this in the lab? There's a couple of different ways you can do so. So if you had an image like this and a social inference task, so if I asked you to judge the emotion of the person, perhaps in a difficult rating scale, how much fear, how much anger, how much sadness is there, you'd be sitting there scrutinizing the face. And one way that we could measure what you're doing is that we could use eye tracking to measure where you're looking on the face to seek out important information. Another way that we can do it that gives us more experimental control is that we set it up so we only reveal to you little bits of the face at a time that your brain has to work with. So the actual stimulus looks something like this. It's like sliding between you and the face a piece of cardboard into which we've punched a whole bunch of little holes. So every once in a while in a certain trial, you see a little bit of an eye or an ear or a nose or a mouth, and now you have to judge what the emotion is. This is quite difficult. What we can do at the end of that, and we typically show you hundreds or thousands of these stimuli, and we pay you very well to participate in the experiment, we add up all those trials, all those little pieces of faces on the basis of which your brain was able to figure out what the emotion was, and we subtract from that all those trials, all those little pieces of faces on the basis of which you were not able to figure that out. And what that allows us to do is to extract the information that your brain uses to make social inferences from a face. So when you're making social inferences from a face like this, where you look and the information that your brain uses looks like these red regions that you see here. Sorry for the scary look. You shouldn't laugh. This is actually a photograph of one of my graduate students, minus the red. So you look at, your brain uses information from these very red regions, in particular the eye regions in faces. There are muscles around the eyes that give them expression. The eyes move around and tell people where you're looking, and your brain makes use of that and looks at the eyes to figure out how a person feels. We've even been able to measure this in the brain, and again, we've been able to link this to the amygdala, that same structure I was speaking about before. In collaboration here with Huntington Memorial Hospital in Pasadena, as well as Cedars-Sinai Medical Center in Los Angeles, we've had the opportunity to work with neurosurgeons who've put electrodes into neurosurgical patients for the purpose of monitoring epilepsy. And we can measure how single neurons in their amygdala respond to these same stimuli I was showing you, these little pieces of faces. And it turns out that neurons in the human amygdala respond preferentially to these red regions, to the eye region in faces. Another piece of evidence for the role of the amygdala in this process is to return to our patient, SM, the one that I showed you previously, who has lesions of the amygdala. And when we use the same kind of approach with her and show little pieces of faces and ask what information from the face does she use in order to make judgments about the emotion, it turns out she doesn't use the eyes at all. So she fails to use information from the eyes that you and I would use normally. So to conclude, the social brain does several things. At the front end, it has to have a very intelligent filter to figure out what's important, where to look, where to seek out social information in the first place. And the amygdala is one of several structures that aids in that process. Once you have that to work with, your brain then figures out what it means by making social inferences. And based on that, it makes decisions on what to do next and how to interact with other people. So, as you can probably imagine, I've only told you a tiny sliver of what we know about the social brain, and that's only a tiny sliver of what we need to know about the social brain. There's a lot more to learn, and ultimately, we want to understand how all these structures work and link this back to the sort of big picture motivating question that I began my talk with. How does this relate to real-life social behavior? That's a huge task. It's very difficult. That's more than any single lab can achieve. And so we have to collaborate with other labs. And indeed, all the contiguous speakers here, Doris Tsao, myself, Antonio Rangel, and Colin Kammerer, whom you hear next, we all collaborate to figure this out, to look at how the brain subserves decision-making and social behavior in humans. So you could think of the level of analysis that I spoke about as asking psychological questions and then using tools from cognitive neuroscience to figure out how the social brain works. But we want to link this to two different levels. We want to go, as it were, down to the level that Doris Tsao was telling you about and ask how this links to single cells, often in animal models. And we want to link it up a level to figure out how people make decisions and how they interact in very complex environments, how multiple brains interact, the kinds of questions that economists and political scientists ask. And that's some of what you'll be hearing about next. Thank you very much.